In anatomy and physiology courses, there can be, such as in the textbook my classes are using, a short chapter that follows the urinary system on fluids. In the chapter that our course is covering, the first part of this brief chapter just quickly talks about where water is located in the body. Some of it is extracellular fluid, some of it is intracellular fluid. The solutes which are in it. Some of the solutes, if they are ions, conduct electricity, which is very important for the nervous system and the muscular system. And then a few words on how we regulate this, because we want to end a day, typically with the same amount of water, the same amount of these electrolytes as we began with. And so therefore there's this balance. Water comes into our body in the food we eat, in what we drink, in metabolic water through dehydration reactions, and water leaves through sweat, through urine, a little through feces. And so therefore we want to balance it. And a number of hormones can promote the migration of water from the kidneys so that we concentrate our urine and lose less water there, and then move it back into the blood. And we do this if we want to conserve water. Hormones like renin, ADH, and aldosterone. Aldosterone allows us to conserve salt, which then helps us conserve more water. Whereas other hormones like ANP, or preventing the release of something like ADH, would then cause more water to migrate from the blood into the kidney tubules and be lost as urine. So we can control and regulate the amount of water in our bodies. In the same way, aldosterone will promote the reabsorption of sodium, but the excretion of potassium. Other hormones, such as calcitonin, promotes the reabsorption. My fault. Calcitonin will promote the loss of calcium, while parathormone will promote the retention of calcium. And so the first part of this chapter, then typically goes just some quick topics on fluids and the electrolytes in them. The second half of the chapter deals with acids and bases. Water molecules can dissociate to form a positive hydrogen ion and a negative hydroxyl ion. And the concentration of these hydrogen ions is what the pH scale measures. At a pH of 7, there is a balance. For every hydrogen ion, the red dot, there is a hydroxyl ion. And when they are in balance, then the pH is at the midpoint in the scale, a pH 7. It is neither acidic nor basic. Anything with more hydrogen ions than this is acidic, and the pH is then less than 7. 5 or 6 would be a weak acid, 1 or 2 would be a strong acid. Anything with more hydroxyl ions than hydrogen ions would be basic. Something that is a weak base would have a pH of, say, 8 or 9, a strong base 13 or 14. Now, this is important for the body because changes in pH is one of the things that can change the shape of a protein. And when proteins change their shape, they don't work necessarily anymore. And this can result in death. And so the body has a very narrow pH range. The blood's pH is around 7.4. If it drops below 7.0 or rises above 7.8, death can ensue, coma, uncontrollable muscle spasms. This is also why certain digestive enzymes only work in the acid of the stomach, and others only work in the slightly basic concentration of the small intestine, because pH changes the shape of proteins. So how do we keep our pH constant? Such small changes can have such serious health consequences. Well, we have 3 mechanisms which are used in the following order: buffers, changing breathing rate, and then finally changing the amount of acid released in urine. A buffer is a system where you have both a weak acid, like carbonic acid, and then its base, such as bicarbonate. Because acids can break apart and produce a hydrogen ion, whereas bases can bind hydrogen ions. And when you have both of these together, you have a buffering system. So for example, there are a number of buffers in the body. There's phosphates inside cells. Many amino acids of proteins can serve as buffers. But the major buffer in blood plasma is bicarbonate. So in blood plasma, there is both bicarbonate and carbonic acid. If all of a sudden more red dots were to appear because of, say, lactic acid in exercise or keto acids in the post-absorptive state, if say, and this, diabetics would be more prone to this, or other sources of acidosis, say you weren't breathing deeply enough. Well, under that condition, bicarbonate would bind the excess hydrogen ions, become carbonic acid. Carbonic acid then becomes carbon dioxide and water. Carbon dioxide comes out the nose. Water can be lost a couple of ways. Now the acid is gone. So we began with too much acid, too many red dots, and now they're gone. And so the buffer has resisted a change in pH. You added acid to blood, but yet the pH did not change. This will continue as long as there is bicarbonate. Bicarbonate doesn't last forever. But in the same way, if you don't have enough acid, so if there aren't enough red dots, the carbonic acid, which is in the blood, can now dissociate and produce the red dots that you need. And so in acidosis, this equation moves towards the right and eliminates the hydrogen ions. In alkalosis, then the equation then can move to the left. So buffers work great. Unfortunately, there are limits. At a certain point, the buffers have been exhausted. But at that point, changes in breathing rate can help to maintain a constant pH. We measure pH in our bodies. The medulla is measuring it in the brain, and there are receptors in both the carotid and the aorta measuring pH there. And if the pH were to drop, so if your blood's becoming too acidic, that stimulates the breathing centers, which will now cause more respiration. By more respiration, I mean you can breathe more deeply and also more frequently. Say during exercise, you're breathing both more deeply and more frequently. So this increased respiration can be stimulated by increased acid, higher carbon dioxide levels, which then cause more carbonic acid, and lower oxygen levels. And so when our blood becomes acidic, we breathe more, and as we exhale more carbon dioxide, that's less carbonic acid, and then the pH, the acid level then drops back to normal. If, however, our blood isn't acidic enough, then instead of breathing more, we breathe less. So for example, I'm at rest right now. I do not want to be breathing like this. If I am, then I'm losing too much carbon dioxide. My blood has too little carbonic acid. I have too little acid. That's a problem. And so under that condition, I would simply breathe less. I wouldn't breathe frequently and deeply. And so low acid levels, instead of simulating increased respiration, would cause a decrease in respiration. And so our second mechanism is that we change the rate of respiration. The third mechanism, this takes the longest to engage, is that we change how much acid can be released in our urine. The kidney tubule cells can take carbon dioxide and water, put them together to make carbonic acid, and then can make hydrogen ions and bicarbonate. The bicarbonate can be shipped into the blood, where it's a blood buffer, so that will make the blood less acidic. And the hydrogen ions can then be pumped into the urine, and thus acid is excreted from the body. So that's a third way that you can control the amount of acid in your body, by changing how much hydrogen ions are excreted in urine, and also then how much bicarbonate goes into your blood. Now, some individuals suffer acidosis, where their blood does become too acidic. And one of the causes for this is if you take a diuretic, and diuretics may interfere with, diuretics can interfere with the pumping of hydrogen. They inactivate that sodium anti-port pump where sodium comes in and hydrogen leaves. And then as a result, we lose this ability to secrete hydrogen, and acidosis can result. Breathing is involved in controlling our pH, and thus anything that interferes with that, such as an airway obstruction or emphysema, or maybe a drug or alcohol overdose, which would affect the breathing centers, that could then eliminate our ability to respond to an acid challenge. Chronic diarrhea can cause the loss of bicarbonate, which is important as a buffer. So there are a number of possible causes of acidosis. There are also causes of alkalosis. I could hyperventilate, so that could be a cause of losing too much acid. And another cause of alkalosis is that if I had continual vomiting. When I make stomach acid, it starts off as carbonic acid, and then bicarbonate is exchanged for chloride ions. The negative chloride ions plus hydrogen go into the stomach, so there's hydrochloric acid in the stomach, but bicarbonate goes into the blood, one of the reasons we become sleepy after a big meal. But if I were to vomit, then if I made more stomach acid, more bicarbonate would go into the blood. If I vomited again, more bicarbonate would go into the blood, and then the blood would become basic. And so that can be a cause of alkalosis. And so this is a short chapter, and half of it is on acids and bases, how we regulate our blood's pH, and then things that can cause acidosis and alkalosis. And as always, this was a quick synopsis of the videos in the playlist, which go into a little more detail.